So welcome again to this channel. So today we discuss the DMLT Examination Board Odisha second year DMLT Annual Examination 2026. ओके सो बायोकेमिस्ट्री का पेपर है, ठीक है, जो कि पेपर 3 का. ओके सो बेसिकली ये आज हमारे लेक्चर जो भी है लेक्चर नंबर 3 है, ठीक है. सो टुडे इज लेक्चर नंबर 3. ओके सो आई विल डिस्कस हियर the rest of the question, यानी कि question number 3, question number 4 and question number 5. okay so question number 1 is discussed in lecture number 1, question number 2 is discussed in lecture number 2, and the rest 3 question, okay, 3 यानी कि 3, 4 and 5 is discussed in lecture number 3. okay so without any further delay, let's start the lecture. okay so the first is here एंजाइम्स इन गैस्ट्रिक जूस. ओके सो क्वेश्चन नंबर 3 का फर्स्ट क्वेश्चन क्या है? एंजाइम्स इन गैस्ट्रिक जूस. ओके ओके सो व्हाट इज द सॉल्यूशन हियर? सो द मेन एंजाइम्स प्रेजेंट इन गैस्ट्रिक जूस आर, व्हाट इज द मेन एंजाइम? दीज आर द मेन एंजाइम which are present in the gastric juice: pepsin, renin, gastric lipase. okay so what is pepsin? So pepsin is the it digests the protein into smaller peptides. okay so pepsin का काम क्या है? is generally digest protein into smaller peptide. protein into smaller peptide, right? okay here is the digest. ठीक है, so protein digest into smaller peptides. And what is the functions of renin? Renin is present mainly in infant case. okay in infants it coagulates the milk protein, यानी कि casein. okay so milk protein is casein. so renin present होता है infants. okay present mainly in infants, which helps in coagulate the milk protein. okay then gastric lipase. Gastric lipase is a digests fat into fatty acid and glycerol. okay it digests fat into fatty acid, ठीक है, fatty acid and glycerol. okay and what are the functions of these enzyme? And these enzyme helps in the digestion of food in the stomach. okay so these are the some enzymes of gastric juice which are helps in the digestion of the food in the stomach. okay so this is your question. okay enzymes in the gastric juice and its answer. Clear everyone? okay then next question, the order of draw of blood sample. okay so the recommended order of blood collection is, first is the blood culture bottle. okay so in blood culture bottle, which tubes are generally, we have citrate tube, यानी कि blue top, the blue top is citrate tube, then serum tube, okay यानी कि red and yellow top, then heparin tube, green top, then EDTA tube, the purple color top, and the fluoride oxalate tube, यानी कि grey top. so these are the vials generally we use in the as a blood culture bottle. okay so what is the importance here? It prevents the contamination of additives between the tubes. Clear everyone? okay then the next question is Rothra's test. okay so what is Rothra's test? okay the principle is Rothra's test is used to detect the ketone bodies in urine. ठीक है, जनरली जो कि Rothra's टेस्ट क्या करता है? इट डिटेक्ट द कीटोन बॉडीज इन यूरिन. ठीक है, व्हाट इज द प्रोसीड्यूर? दैट ऐड द अमोनियम सल्फेट टू यूरिन. ठीक है, अमोनियम सल्फेट टू यूरिन. ऐड सोडियम नाइट्रो पेरोक्साइड सॉल्यूशन, देन ऐड अमोनिया सॉल्यूशन केयरफुली. ओके, सो फर्स्ट क्या करेंगे हम? अमोनियम सल्फेट ऐड करेंगे यूरिन में, देन सोडियम नाइट्रो पेरोक्साइड सॉल्यूशन हम ऐड करेंगे, देन अमोनियम सॉल्यूशन केयरफुली हम ऐड करेंगे. उसके बाद क्या होगा? व्हाट इज द रिजल्ट? द रिजल्ट पार्ट इज that sodium it purple or violet ring, okay, it indicates the presence of ketone body. Okay, we saw that purple color or violet color ring are produced that means which indicate the presence of ketone bodies in urine. Okay, then what is the clinical significance? The clinical significance is it positive in diabetes mellitus, starvation, and diabetic ketoacidosis. Okay, so these are the Rothras test. Okay, so here question number 3, it's generally all are 2 mark. ठीक है, इसलिए हम कम ही लिख रहे हैं इसमें. ठीक है, इट जनरली ऑल आर 2 mark क्वेश्चन. ठीक है. Okay. The next question is that preservation of urine sample. Okay, your next question is preservation of urine sample. So commonly preservatives used in urine sample are toluene, thymol, formalin, boric acid, hydrochloric acid. Okay, these are the preservative are used in the urine sample. Okay, and what is the purpose? Why it preserve? Because it prevent the bacterial growth and preserve chemical constituents of urine. It prevent the bacterial growth generally. Okay, okay, I will erase all the ink on this slide, the previous slide also. Just wait a second, so that you take a screenshot. Okay. Okay, clear everyone? Then the next question is creatinine clearance test. Okay, the question is creatinine clearance test. All are 2 mark question. Okay, all are 2 mark question. So what is the definition? That creatinine clearance test measure the ability of the kidney to filter the creatinine from the blood. Okay, so what is clearance test? What is creatinine clearance test? Creatinine clearance test measure the ability of the kidney. Okay, it measures the ability of kidney which to filter the creatinine from the blood. Clear? What is the sample required? Sample generally required in 24-hour urine sample. Okay, and blood sample for serum creatinine. What is the normal value? In case of male is 90 to 140 ml per minute, and in case of female it 80 to 125 ml per minute. And significance is it access the glomerular filtration rate. Okay, GFR, and it used to evaluate the kidney function and diagnose the renal disease. Okay, and diagnose the renal disease. Clear everyone? Okay, then so wait for a minute, I will erase all the ink on this slide. Okay, then the next is, so here sorry, your question number 3 will be end here. Okay, then the question number 4. Okay, so ये जनरली 1 मार्क का क्वेश्चन है. ठीक है, so पूछा गया है, पहला क्वेश्चन क्या है कि टू टेस्ट टू डिटेक्ट द प्लाज्मा ग्लूकोज. ओके, so which two tests are generally occur to detect the plasma glucose. So what is the two test? पहला हो गया जीओडी पीओडी मेथड, यानी कि ग्लूकोज ऑक्सीडेज एंड पेरोक्सीडेज मेथड. दूसरा हो गया हेक्सोकाइनेज मेथड. ओके, सो दीज आर द टेस्ट टू डिटेक्ट द प्लाज्मा ग्लूकोज. क्लियर एवरीवन? देन नेक्स्ट, टू टेस्ट टू डिटेक्ट द सेरम कोलेस्ट्रॉल. व्हाट इज द टू टेस्ट टू डिटेक्ट द सेरम कोलेस्ट्रॉल? सो पहला हो गया CHOD पाफ, यानी कि कोलेस्ट्रॉल ऑक्सीडेज पेरोक्सीडेज मेथड. ठीक है, कोलेस्ट्रॉल ऑक्सीडेज एंड पेरोक्सीडेज मेथड, इन शॉर्ट CHOD पाफ. ठीक है, PAP. Next we can talk about the second test is Lieberman-Burchard method. Okay, these are the two tests to use the serum cholesterol. ठीक है. Then next question, two serum electrolytes. We know the electrolytes that we have sodium and potassium. ठीक है, so here तीनों question solve हो गया. ठीक है. Then the next question, two test for hepatitis. So पहला हो गया क्या, हेपाटाइटिस बी सरफेस एंटीजन, HBsAg, यानी कि उसको हम HBV भी कहते हैं, HBV. ठीक है, जस्ट वेट सेकेंड, HBV. ठीक है, हेपाटाइटिस बी वायरस यानी कि HBsAg, ओके, हेपेटाइटिस बी सरफेस एंटीजन टेस्ट. नेक्स्ट इज एंटी HCV एंटीबॉडी टेस्ट. ठीक है, सो दीज आर द टू टेस्ट फॉर हेपेटाइटिस. नेक्स्ट क्वेश्चन, टू टेस्ट फॉर यूरिनरी ट्रैक्ट इन्फेक्शन UTI यानी कि यूरिनरी ट्रैक्ट इन्फेक्शन. सो व्हाट इज द टू टेस्ट? दैट यूरिन माइक्रोस्कोपी एंड यूरिन कल्चर एंड सेंसिटिविटी टेस्ट. ठीक है, सो हियर योर क्वेश्चन नंबर 4 इज एंडिंग. ठीक है, देन द नेक्स्ट इज क्वेश्चन नंबर 5. ये जनरली 0.5 मार्क क्वेश्चन है. ठीक है, यानी कि दो मिलाकर 1 मार्क होगा. ठीक है, सो द पहला क्वेश्चन इज, the first question is here, the end product of glycolysis. What is the end product of glycolysis? Is pyruvate or pyruvic acid. Clear? Next question, the normal fasting glucose level is, what is the normal fasting glucose level? That's 70 to 100 mg per dl. Okay, we know that 70 to 100 mg per dl. Okay, so actually it vary in some cases. Okay, 70 to 100 or 70 to 110. Okay, it generally vary. Okay, so but the average is 70 to 100 mg per dl. Clear? Then the next question is, the enzyme used as a cardiac marker. Okay, the which enzyme is used as a cardiac marker? So the answer is here CKMB. That means creatinine kinase MB. Okay, CK means creatinine kinase MB. Next question is, clear everyone? The normal range of serum potassium. What is the normal range of serum potassium? Serum potassium का normal range कितना होगा? 3.5 to 5.0 और somewhere 5.2 to 5.5 milli equivalent per liter. Okay, it, I will already say that it's somewhere it vary the normal range. Okay, but the average is 3.5 to 5.0 milli equivalent per liter. Clear everyone? Next, the hormone secreted by the thyroid gland. Which hormone is secreted by thyroid gland? The answer is T4 यानी कि thyroxine. Okay, so the answer is here that T4. Okay, so T4 is the answer, thyroxine. Then the next question is, the major protein present in plasma. So which is the major protein that present in plasma? The answer is albumin. What is the answer? The answer is albumin. Okay, albumin. Then the next is, the enzyme elevated in acute pancreatitis. So the enzyme elevated in acute pancreatitis is serum lipase. Clear everyone? Then the next question is, the pigment responsible for jaundice. So which pigment are responsible for jaundice? The answer is bilirubin. Okay, bilirubin is the answer. Next question, the anticoagulant used in blood glucose examination is which? So which anticoagulant is used in blood glucose estimation? That sodium fluoride potassium oxalate. Okay, next question, the hormone that lower the blood glucose level is insulin. Okay, so which hormone that lower the blood glucose level? The answer is insulin. Okay, clear everyone? So here your all the question is completed. Okay, so we completed question number 3, 4 and 5. Okay, so this is all about your biochemistry 2026 annual examination, the second year question. Okay, I hope you understand all the answer and all the concept here. Okay, so thank you.